I'm going to also post my day's date from Sam's review that she previously gave on Saturday. QR code or using the... And then without further ado, we will start our review. Or do we have any questions? Cool. Okay. So we got our review. Oh, okay. So first, we just go over basic anatomy or like the general anatomy of the brain. So we have our 4 basic lobes of our cerebrum and our cerebellum and our brainstem down here with the pons, medulla, brainstem. Midbrain is inside mid. We have our central sulcus, and then we move, and then we send, and parietal morbials are on our cerebellum. There's also another way of looking at the brain and breaking up the different sections. So one is based on lobes, and then we also have Brodmann areas where we can go a little more detailed. And here you can see the central sulcus, and then around it we have primary motor, primary sensory, and then the different areas. So we have primary visual, and then the associative areas. We'll have Wernicke somewhere over here, Broca somewhere over here, closer to motor, closer to understanding when we talk about hearing and aphasia and auditory. I don't know if your professor really wants you to know any of the specific Brodmann area numbers. Does he really specify which ones, or not really? Does he talk about it? Maybe mention it? Sometimes mentions it, but maybe doesn't ask specific questions, which is nice, because there's a lot of areas. Because I took him for neurobio, I took Chang, not Bossy. I took Chang for neurobio, but I also had him for like molecular and cellular basis of brain function, so they kind of interconnect when I think about them. But then we also have the interior view, or the ventricle view. So we can see our lateral ventricles that go through the cerebral hemispheres, and then we have our 3rd ventricle and our 4th ventricle down near the pons and the medulla. So when we talk about these areas, we kind of know where they are. Okay. Then if we look on the underside of the brain, we have our cranial nerves, and these is kind of a mnemonic to tell you about the order. This is on, they traveled and found Voldemort guarding very ancient Horcruxes. On top, fin and germ, just information. Get the order of your cranial nerve, trochlear, glossopharyngeal, and then VA, and then hop up. It's good to take note of this is kind of to the right, a saying that helps you remember if they're sensory motor or both sensory motor. And it kind of matches up with the number. So this one says, some say money matters, but my brother says big brains matter more. So that's one way of saying. So anything with an S is just sensory, so olfaction, ocular, optic, sorry. Factory, optic, just sensory, and then they have just the muscular, just motor, like oculomotor and trochlear. And then the B's are both. So that's one way of memorizing them, to kind of get you a refresher on what their functions are. Okay. Or do we have questions so far on cranial nerves or anything like that? Yes. I think the final's cumulative, if I'm not mistaken, right? Yes. Oh, okay. Then probably not the same exact, but I would say similar. Okay. Then it's more cumulative than the other one. Yeah, so 3 was probably cumulative up to that point, and then the final is like, here, add some more.
If we did like 3/4 of the final already, we do the same as 3 of like 6 chapters.
Okay. Okay, not so bad. So you've had like enough practice with like some of the beginning stuff, so it's not too bad. Okay. I'm not like completely sure and up to date with what's happening in the class because I see new faces. Some of you haven't gone to tutoring. No, but like that's kind of what helps us figure out where you guys are, what to focus on, how to make these reviews a little bit better is when you attend. But I guess we just need to advertise a little bit more. So we did not do very well with that. But oh well. Next, we can look at the spinal cord. So this is another way of remembering which side is looking at motor information going out and which side is sensory going in. So there's VEMDOS, ventral efferent motor. So your ventral root is for your motor information and going out to your body. And your dorsal side is the sensory going back up to the brain, or if it's part of a reflex, then just the spinal cord, but toward the CNS versus away from the CNS. I usually don't use VEMDOS. I usually just think of dorsal root ganglion, and there's only needs to be a ganglion for sensory because that's where like all the cell bodies are to get information from the body and then send it away. But so it's whatever will help you remember which way is what. So I usually just think of the sensory, the dorsal root ganglion, and think, okay, well that's going to be sensory, that has a ganglion. Versus the ventral root, which doesn't have a ganglion. It's just the neuron axons. Okay. And then with like when you compare the spinal cord to the brain has its gray matter on the outside and its white matter, basal ganglia on the inside. So the white matter is the myelination, and the gray matter is usually those cell bodies. But in the spinal cord, it's flipped, where the white matter is on the outside, the gray matter is in that horn area. So that's one thing to note as well, like difference between spinal cord versus brain. We have a difference in where the white matter and the gray matter are. Okay. So one question is, did I go over it? I went over some of this, maybe. So question one is, which of the following provides sensory information for vision? So provides the sensory information, so brings it to the brain, because it's taking sensory information and bringing it to the brain. So A says the dorsal root of the spinal cord. Do you agree with that? Why or we're not? Oh, there isn't all of the above answer, that's why. B says occipital lobe of the cerebrum. C is cranial nerve 2. And D is Brodmann area 17, 18, and 19. So if we're providing sensory information for vision, if we're getting it, if we're retrieving it and sending it to the brain, that's going to be not through the spinal cord, because sensory information for vision is going to be going through the optic nerve. So the optic nerve is cranial nerve 2. It's not going to be the occipital lobe of the cerebrum, because that's not where we're getting the sensory information. That's where we're interpreting it, and that's where we're kind of piecing it together. That's what the brain is doing there. And then Brodmann area 17, 18, 19 would be part of the occipital lobe, but it's again where the information is processed, not where you get the information. So you're not sensing anything in your brain, you're sending things from your senses to your brain, and you're doing that based on the optic nerve, cranial nerve 2. So for this one, it would be C. Do we have questions on that versus bringing it in versus where you're processing what's going on?
What did you say about the dorsal root?
For the dorsal root of the spinal cord, that is where sensory information comes from, but it's not for the vision. Vision goes through the cranial nerves, yeah. So like for something else, if you were like touching something hot or felt pressure somewhere, like something from your body, that would go through your spinal cord, through the dorsal root. But since this is vision, this is one of those special senses that has a cranial nerve specifically for it. So we don't go through the spinal cord. Occipital lobe? The occipital lobe is where vision is interpreted, not where we're sensing vision. And it's asking provide sensory information. So you're not sensing in the occipital lobe. You're processing what you sensed. Okay. Okay. So now we can look at types of neurons. So the majority of the neurons in your brain and your central nervous system are these multipolar neurons. We also have bipolar neurons, which are usually the special sensory ones. So we have like the retinal one that we'll see if we look at the eye, and we'll have the olfactory nerve as well, neurons as well, for olfaction. Then we have unipolar, which is where we'll find those in the spinal cord. So like the cell body you see on a unipolar would be in that dorsal root ganglion. We'd have some of them going out to meet with bipolar neurons, and then some of them going in to synapse in the spinal cord, or all the way up into the brain. Okay, and then besides neurons, we also have glia cells or glial cells. So these are support cells. They're going to help our neurons survive in the CNS and PNS. So central nervous system, peripheral nervous system. A mnemonic to remember the glial cells for central nervous system, we have omega. So O is oligodendrocyte, M microglia, E ependymal. G is just glia, because we needed omega, it sounded weird. And then A for astrocytes. The PNS gets Schwann cells and satellite cells. They don't really talk about satellite cells usually that much, but they exist and they're in the peripheral nervous system. And I think that's usually all you need to know about those at least. But the other ones, their functions are important. So like Schwann cells are going to be for what? Does anyone remember their function? Myelinating. So PNS, the Schwann cells are for myelination. And the CNS, what does the myelination? Oligodendrocytes. So oligodendrocytes are for myelination in the central nervous system. Peripheral nervous system has those Schwann cells. So the difference between like an oligodendrocyte and a Schwann cell would be their location, and also like when you have one Schwann cell, it can only help myelinate one neuron, because one neuron will have a bunch of Schwann cells attached to it, kind of like beads on a string is usually the analogy they give. But for oligodendrocytes, one oligodendrocyte is going to myelinate many axons and many neurons. So it's going to kind of act like a web, or like a spider, or like a star type thing. Even though we call astrocytes the star ones, oligodendrocytes kind of look similar, but they're myelinating. And they're attaching to multiple axons from multiple different neurons. That's why Schwann cells are usually involved in helping a neuron regenerate if we have something that breaks in the CN and in the PNS. So it can have a chance at regenerating because it's just a bunch of Schwann cells help one axon, but in the CNS it's harder because oligodendrocytes are just for many neurons and they don't really keep track of their own axon and what happened. They also don't have like this type of neurofactors to release for that. But part of the reason is also like how they myelinate, one versus many. Okay, microglia are for immunity. They are kind of like macrophages in the CNS because the actual antibodies and things like that from your immune system can't get into your brain, which is good, but it could be bad if there is something bad in your brain. So you have microglia for that. Ependymal cells are helping to make cerebral spinal fluid. They also have those cilia that are like moving the CSF through the ventricles and down through the spinal canal area, or central canal of the spine, of the spinal cord. And the astrocytes kind of do a lot to list. They help make sure external concentrations of ions are kept at a certain level so that neurons can function. They help form the blood-brain barrier, so they take in nutrients from the bloodstream and then they help give it to the neurons. So like neurons usually can't use glucose as is, so astrocytes usually metabolize it to something that neurons can use. And then astrocytes help with, I think, potassium concentrations. It can help with picking up neurotransmitters that are kind of released in synapses near it, so it kind of helps clean up and organize everything. Usually, like, if it's not one of the things that everything else does, usually astrocytes do almost all of it. When in doubt, astrocytes. But do we have questions on glial cells? Are we okay? No questions. Cool. Okay. Then we have a picture depicting our glial cells. So over here, lining our ventricle, we have these ependymal cells. Over here, we see oligodendrocytes because they're myelinating a bunch of different neurons or axons. And then over here, we have that astrocyte clinging on to the plasma or the capillary, and then clinging on to a neuron to try to be that bridge between it, forming the blood-brain barrier. Or it also helps like form it with these cells of the capillaries, because they kind of fill in the cracks and keep things from being able to slip out easily between cells and diffuse. And then, yeah, we have a microglia here. It's smaller than all the others, hence microglia, very small cell. And then I think those were it. Yeah, those were it for the CNS, because like the satellite and Schwann cells aren't going to be found in this type of picture. You're not really going to see CNS and PNS combining in the same picture. Unless they're showing two different parts of the body, of course. So yeah. Okay. And then we have questions. So it says, which of the following is a function of astrocytes? So A says it helps produce CSF. B, it says it helps form the BBB, or blood-brain barrier. C, it is involved in the active immune defense in the CNS. D is myelination in the CNS, and E is none of these. Okay, so what do we like? What answer choice? We like B because it helps form the blood-brain barrier, which is true. A helps form the CSF is for what kind of cell? What kind of glia? Ependymal or ependymal or ependymal, yeah. Those ependymal cells are for producing that CSF. What about the active immune defense in the CNS? Microglia or microglia. I think we usually say microglia, but you hear many. As long as you can notice or see the word and know that it's what you're talking about, you're fine. Because they're not going to have you do a free response, spell it the correct way type thing. Demyelination in the CNS is what kind of cell? Oligodendrocyte. And of course it's not none because it's B. Then we can start talking about the membrane potential of a neuron. So I kind of tried to draw a membrane with the different receptors. So I just made them different colors and different shapes to emphasize that they have different, not receptors, but different channels for them. Because in order to have a membrane potential, in order to have that gradient, you need a semi-permeable membrane. If you don't have a semi-permeable membrane, you can't really have a gradient of chemicals or electrical particles. So in order to have that electrical chemical gradient or a gradient in general, you need a semi-permeable membrane so that your particles can move from higher to lower or lower to higher. Okay, so we have a chemical gradient because we have higher concentration and lower concentration of our ions. So potassium's high inside the cell, low outside, and then sodium, calcium chloride, or high outside the cell and low inside the cell. So if they want to go down their gradient, down their concentration gradient, potassium would flow out, sodium, calcium, and chloride would flow in, going down their concentration gradient. Because of this concentration gradient, it kind of creates a more negative charge inside the cell and a more positive outside the cell. Not just because of this, but mainly because of the sodium-potassium ATPase, where for every, I think, 2 potassium that gets thrown inside, 3 sodium get thrown outside. So if you keep having 2 positive charges put inside and 3 taken out, you're going to have a net positive inside or a net negative out. Negative out. Reverse that, sorry. My bad. So whenever you put 2 potassium in or 2 positive charges inside your cell and take 3 sodium out, you're having a net positive outside your cell or a net negative inside. So because we have that net negative inside, it's going to attract things that are positive and repel things that are negative. So if potassium is going down its electro gradient or electrical gradient, it would be going inside of the cell, which is contrary to its concentration gradient. But sodium and calcium would be going inside the cell, whether it's their chemical gradient or their electrical gradient. Chloride would be leaving if it's going based on its electrical gradient, because it's negative and in the inside of the cell it's negative. So for potassium, usually when potassium channels open, it flows outward. It's going based on its chemical gradient, not its electrical gradient. And sodium, when it goes inside the cell, when the channels are open, it's going down both its electrical and chemical gradients, because it's positive going toward the negative and it's high concentration going to low concentration. So it's kind of important to take note of. Okay. Do we have questions on electrical chemical gradients? You okay? Okay, we're cool. Then if we want to calculate the potential of our cell based on individual ions, so if we just want to compare potassium to itself, like we can use the Nernst equation. That helps us mainly to kind of see what the membrane potential would be if the membrane was only permeable to that one ion. So this is like, Nernst equation is for each individual ion. And then we have Goldman's equation, which helps you calculate the membrane potential overall based on all of the ions interacting. So Nernst equation is for one ion, Goldman equation is for the overall membrane potential. If you need to use it, I'm not sure if he's going to have you calculate things on the test, probably not. But it's okay. But good to make note of like when to use it and what it's for. Okay, then we have another question. So question 3 is, which of the following will occur when the potassium ion channels of a neuron are open? A says potassium ions will enter the cell. B says potassium will flow down its electrical gradient. C says potassium will flow down its chemical gradient. And D says the neuron will depolarize. And then E says all of them will happen. Okay. So we're talking about potassium channels that open. So where is potassium highest, inside the cell or outside the cell? Inside the cell. So if it's going down its concentration gradient, where would it be heading? If it's higher inside than outside, where's it head if it goes down its concentration gradient? Heads outside. Okay. So which will occur when potassium channels open? So usually when potassium channels open, potassium goes down its chemical gradient. If it goes down its electrical gradient, it would be going inside the cell because potassium is positive, inside is negative. But usually when we open potassium channels, it flows out. It goes down its concentration gradient first, and then it will slowly kind of be pulled backwards when it kind of reaches equilibrium, if it reaches equilibrium, if it gets to that point, because of the pull of its electrical gradient. Potassium does not enter its cell. It doesn't really flow down its electrical gradient because those are kind of the same thing. So flowing into the cell or going down its electrical gradient is the same. And it doesn't do that when we open potassium channels. We'll see like with our, what's it called? With our action potential, that's not what happens when we open potassium channels. Also, when we look at neurotransmitters and transduction, that's also not going to happen if we open potassium channels. So potassium's not going to enter, it's not going down its electrical gradient. It will go down its chemical gradient, and then the neuron will not depolarize because the potassium will be leaving. So positive is leaving, and it's already negative. So it's negative inside the cell, and now we're taking positive things out of it. So we're becoming even more negative. And what's that called when our cell gets more negative or goes below resting membrane? Hyperpolarization. Yeah. So it's not going to depolarize, it's going to hyperpolarize. So for question 3, the answer is going to be C. Potassium will flow down its chemical gradient, which is flowing outside of the cell, or hyperpolarizing the cell. Okay. Then we have a graph of our action potential. So resting is around -70 or -65, some say. It depends. Then we have our action potential, our rising phase, which is our depolarization. So around here is when our sodium channels are opening. So they're all the way open until we get to the very top, and that's when our sodium channels will close. And our potassium channels are open at that point. They start opening a little bit earlier though, but they're very slow to open. So potassium's slow to open. So by the time it gets to the apex, when sodium closes, potassium will be fully open. And then we're going to have that hyperpolarization or repolarization. Yeah, they usually call it depolarization or repolarization. So when it dips below, that's when we start calling it hyperpolarization. So we have depolarization from sodium channels opening, sodium channels closed, potassium is now open. We have that repolarization, and then we have that hyperpolarization when our potassium is going to close. So then at this point down here during hyperpolarization, sodium potassium channels are closed. We have that sodium potassium ATPase trying to get our resting membrane potential back to normal. And that's why it goes back to rest eventually. So during this time, we have the absolute refractory period. So once our sodium channels close and our potassium open, or during the sodium channels still being open, they can't open again until a little bit has gone by, and then we'll get to our relative refractory period over here, when we can have another action potential. Okay. And then our action potential is all or none. So we aren't going to have some graded like bigger than 30 or lower than 30. It'll be around 30, but it'll be consistent in every neuron. It's limited by, so the upper bound is limited by the sodium solubility, or not solubility, potential. So if the membrane was only soluble to sodium, that's kind of where the maximum lies. And then the minimum lies if the membrane was only soluble to potassium. So the solubility, not the solubility, but the membrane potential for sodium versus potassium are kind of the bounds for this, because we have sodium opening and then we have potassium only open when their sodium is closed. So it kind of falls in that area on the graph. But yeah, action potential is all or none. Okay, and then we have a little bit of information about action potential propagation. So when we increase the diameter of the axon of our neuron, we're going to decrease the resistance for things to flow along the axon to go down it. So it's kind of like having a hose with a bigger diameter than something with a very small diameter. It's very easy for more water to flow with the bigger hose versus the smaller one. So that's why we want to decrease resistance along the axon. So we want things to flow better down. And then myelin increases resistance across the membrane or across the axon. So it's keeping kind of everything from leaking out. It makes it faster and more efficient when we have myelination versus when we don't have any myelination. So when we have myelination, we only have really the ion channels being able to open at those nodes around the air, and then kind of jumps from node to node and goes down our axon much quicker than going for every single piece of the axon all the way down. Okay, so we want to decrease resistance along the axon, but increase it across the membrane. Because we want to keep everything in, but we also want to allow things to move down well and efficiently. So increasing diameter of the axon and then myelination helps with that. Question 4 says, which of the following is true during an action potential? Not an actional potential, actually, but an action potential. We'll just ignore that part. A says, neurons with unmyelinated axons have more sodium channels, which produces a larger action potential. Do we agree with that? No. So although they may have more sodium channels, they're not going to produce a larger action potential, because action potentials are all or none. There's only one magnitude. You can't have varying degrees of an action potential. So that one's bad. B said sodium channels are open during repolarization and closed during hyperpolarization. That's not true. During repolarization, our sodium channels need to be closed so we can actually get the positive ions out. We want to go back to negative during repolarization, or to positive. So Sodium channels are actually open during the depolarization part, and then they are closed during repolarization and usually during hyperpolarization as well. So potassium channels would be open during repolarization and closed during hyper, or they close during hyper. Okay, so B is not good either. C says myelinated axons have a lower resistance across the membrane, which allows for a faster action potential. So parts of that is true. So myelinated axons do allow for a faster action potential, so go down quicker, but they don't have lower resistance across the membrane. They have a higher resistance across the membrane, so things don't leak out. So it's like myelination is kind of like tape around a leaky hose, keeping everything in. So they increase the resistance across the cell membrane, keeping everything in, and that makes it faster, not a lower resistance. So C is also not good. Our answer is going to be D, none of these, because none of these are good. Be good with that. Okay. Then we can talk about, now we know about action potentials, we can talk about neurotransmitter release. So these are the 4, I guess, main steps. I broke them into steps of our neurotransmitter release. We have that action potential, which is the main thing that sets us off. So we have action potential, and then that action potential is going to cause a depolarization in our cell. That depolarization is going to activate these voltage-gated calcium ion channels, and those are going to open. So voltage-gated calcium ion channels mean they open because of a depolarization or because of some charge change. That's what the voltage is meaning. So it's voltage-gated, so it's open based on a charge change, and action potential is a depolarization or a change in charge. So calcium channels can open. When calcium channels open, where does calcium flow? Inside the cell or outside the cell? Remember? Anyone remember? Calcium, where's it flowing? It's going in. Because calcium's higher on the outside, lower on the inside. For its chemical gradient, it's also positive and the inside is negative. So it also goes down its electrical gradient. So it's being pulled like twice as hard. So voltage-gated calcium channels open. Calcium goes in our cell. The calcium's gonna interact with the SNAP proteins, or I think they call SNAPs or synaptic proteins, but they're going to interact with those proteins which are holding the vesicles and docking them to that activation zone. So those docked vesicles are then going to, the proteins docking them are going to interact with calcium, and they're going to exositize those vesicles, and those vesicles are going to be holding those neurotransmitters. Then those neurotransmitters are going to be released into the synaptic cleft. So those are our steps, and then we can see it with a picture is over here. So we have our action potential coming down, our calcium channels opening on our presynaptic membrane. Calcium's flowing in. Calcium is forming a protein complex with the proteins docking the vesicles. They're called snare complex, or sometimes they're called SNAP proteins. Or I guess proteins is repetitive there. That's okay. So snare proteins or SNAPs, I think is what they're called. And they're docking those vesicles filled with neurotransmitter. Calcium interacts with them, forms a complex, they exositize, and then neurotransmitters are released. So that's what's going to happen whenever we have neurotransmitters are being released, usually for all neurotransmitters. It depends, though, on those special sensory ones when they release neurotransmitters, because they're a little bit different. But these are for the regular, normal neurons, not in the sensory system. So this is like what happened in CNS stuff. So action potential, calcium channels open, calcium flows in, forms a complex with the docked vesicles, the vessels exositize, and neurotransmitters are in the synaptic cleft. Okay. And then this is what happens when our neurotransmitters interact with receptors on that postsynaptic membrane. So over here we have presynaptic membrane, and we have postsynaptic membrane, and we have the synapse. So the presynaptic membrane is releasing neurotransmitters. Postsynaptic membrane is going to have the receptors for those neurotransmitters. So those receptors can either usually cause a depolarization effect or a hyperpolarization effect. So a depolarization effect would be excitatory. So that'd be an excitatory postsynaptic potential, or a EPSP. Something that makes an inhibitory effect would have an inhibitory postsynaptic potential, or an IPSP. So something that is going to hyperpolarize the cell, like potassium leaving or chloride getting in, is going to cause an IPSP. So something that's going to stop a action potential from occurring in that postsynaptic neuron. And then something that's causing a depolarization effect, or an increase in positive charges inside the cell, is going to make an EPSP, an excitatory postsynaptic potential. So like sodium channels opening, sodium rushing in, or calcium rushing in would be an EPSP type event. So those are going to make it more likely for that neuron to then fire an action potential. Then with these EPSPs, IPSPs, we have the synaptic integration, so things that are nearby, or so that would be spatial. So when we have things that are near each other, so two neurons sending neurotransmitters to one other neuron, those would be summed. If they had like EPSPs, they would sum their EPSPs into something bigger. Or if one neuron is just firing really fast, that would be more temporal because of the rate it's firing, and that would summate as well. Then we also have inhibition, so we can shunt it, so stop the neuron with another neuron from firing, and we can send those IPSPs in other areas to kind of stop an action potential from forming in the other neuron. I should have had a picture with that, it would be better, but it'll be okay. Okay, so question 5 says, if voltage-gated calcium channels are blocked, what would happen in the synapse? So A says, voltage-gated sodium channels would open instead of voltage-gated calcium channels. Is that how that works? Do we just decide to open different channels? Answer is no. So if calcium channels are blocked, then they are blocked, and we don't really have a mechanism to get around it. It's just something bad happened. B says the postsynaptic membrane would hyperpolarize. So the postsynaptic membrane probably wouldn't have anything happen, because if we look back and we didn't have our calcium channels open, or they're blocked, that's what happened, they were blocked, we're not going to be releasing neurotransmitter. So if we're not releasing neurotransmitter, then nothing's going to be happening on that postsynaptic cell. Okay? So the postsynaptic membrane really wouldn't have anything happen to it. It wouldn't be hyperpolarizing or depolarizing. No neurotransmitters would be released because no calcium channels were open to allow calcium to release them. C says that less neurotransmitter would be released. Kind of. It's more like no neurotransmitters are being released. It's not just less of them are getting out. We need calcium to release them. There is no calcium getting in. They are blocked. So it's really none of these. We're not just decreasing neurotransmitter. We're not releasing any of it. So the other options were all of the above and none of the above, and the answer is E, none of them. They're all wrong. Okay. Then we can look at some stuff about the sympathetic and parasympathetic nervous system, or it's part of the peripheral nervous system, specifically part of the autonomic nervous system. Because we have, in a peripheral, we'll have autonomic, we have somatic, which is the actual, like, moving your muscles and things like that, things you have a control of. We have the autonomic. There's also a third one that's mainly for digestion, but no one really talks about that, and we're not really going to talk about that either. So this is the autonomic nervous system section of your peripheral nervous system. Specifically, that's broken up into parasympathetic and sympathetic. Or maybe, okay, anyway, it's okay. Anyway, so parasympathetic is usually shortened to rest and digest. So that one's when you're more relaxed and good things are happening. Sympathetic nervous system is that fight or flight kind of anxiety, running for your life type of reaction. So we're going to look at kind of neurotransmitters that are involved in both of these reactions. Or not reactions, but both of these states of your body. So acetylcholine is one of the important neurotransmitters actually for both of these. So if we actually look back at this picture, the sympathetic nervous system will first come from our thoracolumbar region, and it will synapse in our sympathetic chain and then go to the effector cell. And then in the parasympathetic nervous system, we're going directly from the craniosacral regions and going to the effector cell. They have a synapse here. So we say the neurons coming from the spinal cord are called the preganglionic neurons, and then the ones that are either in the sympathetic chain for sympathetic nervous system or near the effector cell are called postganglionic neurons. So between postganglionic and preganglionic, we have acetylcholine being released in both of these systems. So going from neurons in the spinal cord to either the sympathetic chain or the spinal cord to these effector cells, we have acetylcholine being released, and we have cholinergic receptors to receive them on that postganglionic postganglionic membrane. So specifically, you have nicotinic on that postganglionic membrane in both sympathetic and parasympathetic. And nicotinic are those inotropic receptors, so they're going to be opening ion channels. So in these synapses, we have acetylcholine being released. It goes to a certain receptor, and that receptor is opening ion channels. The other type of cholinergic receptor or receptor for acetylcholine is muscarinic, and that's going to be mainly in the parasympathetic nervous system, and that's going to be between the postganglion and the effector cell. So in the parasympathetic, it's going to be over here by this small postganglion, and the actual thing it's affecting, whether it's the heart or the lungs or digestive tract. We also find nicotinic in the neuromuscular junction, so between nerves and actual skeletal muscle. So they have another area that they're used as well. Okay, so acetylcholine is also shortened to ACH. So be familiar with that shorthand version. It's made from acetyl-CoA, which is this molecule here. You don't really need to know exactly what the molecules look like, but I thought it'd be helpful to get a good visualization of what's going on, or just to scare you with organic chemistry, either one. So this is acetyl-CoA, and then it combines with choline. It combines to make acetylcholine with this enzyme, choline acetyltransferase, or CHAT. So CHAT is the enzyme that helps form acetylcholine. Acetylcholine is an amine, so it's made in the it's made in the terminal button, or the axon terminal. So usually amines and amino acids are made in the terminal. Things that are peptides are going to have to be made in the soma of the cell, or of the neuron, because peptides are kind of like proteins, and they need ribosomes and things like that to be made. Okay, so these are amines, so it's made in the axon terminal. It's made by combining acetyl-CoA and choline and this enzyme, choline acetyltransferase. So this enzyme is a protein. All enzymes are usually proteins, and that was made in the soma, and it traveled down the axon to the terminal. So that's how you create it. And then ACH degradation, this is occurring not in the axon terminal, this is occurring in the synapse. So after acetylcholine is released, it binds to a receptor, it then falls off, and then it gets degraded by acetylcholine esterase, so ACHE, or ACE. I don't know, you can call it that if you like. So then it's degraded into acetic acid, which is over here, and this choline. So you'll notice that choline is used to make acetylcholine, and choline is what you get when you break it up. So this choline is going to be taken back up into that neuron and used again to make acetylcholine. They're going to recycle it, kind of. So they're going to pick this up and make it again when they break acetylcholine down. So acetylcholine, one of the receptors was nicotinic receptors. So this is a transmitter-gated ion channel, so it needs acetylcholine to bind, and then the ion channel will open. So that's what it means, transmitter-gated. So it just means the gate's locked unless you put a neurotransmitter in, that type of thing. So specifically, transmitter-gated ion channels are ionotropic. And again, these are the ones that are found at the neuromuscular junction, not the muscarinic ones. Nicotine is kind of what it's named from. That's an agonist of this, so it acts like it can bind and cause the reaction that acetylcholine would bind and form. And then curare is an antagonist. It does the opposite effect. It kind of just blocks the channels without producing the good effects, which kind of makes it do the opposite. So if, yeah. Ooh. And this is like what nicotine looks like and plant it comes from, and then this is what curare looks like and what it comes from, just for fun. Okay, so with our nicotinic receptors, we're going to have that action potential on our preganglion, if we're talking about this autonomic nervous system. So our preganglion will have an action potential, voltage-gated calcium channels will open, calcium will interact with the proteins and cause synaptic vesicles to exositize, and transmitters will be released into the synaptic cleft. Then the acetylcholine will bind to the ligand-gated sodium channels. Potassium also kind of exits through the same channel, but the main effect is going to be by the sodium. Sodium is going to go inside the cell or outside the cell when sodium channels are open. Inside. Sodium channel is going to rush. So sodium is going to rush inside the cell. It's going to cause a depolarization because sodium is positive. The inside of the cell is usually more negative. So it's going to cause a charge change, a depolarization. Yeah, so sodium flux causes depolarization. And that's kind of where we stop with that kind of information. And it's really going to have other kinds of effects. So if it's on the neuromuscular junction, this sodium influx depolarization is probably going to open more calcium channels, and then calcium is kind of what's helping to contract those muscles. And this is kind of a picture representation of what the neuromuscular junction looks like. So one of the examples of where you would find nicotinic receptors. So you have your neuron, has an action potential, calcium flowing in, allowing acetylcholine to be released, acetylcholine binding to its receptor. It'll cause so when the receptor opens or the channel opens, sodium rushes in, potassium kind of comes out, but not as much. And then you're going to have your acetylcholine esterase breaking acetylcholine down into acetate and choline. Choline is going to be taken back up into our neuron. Okay. Do we have questions so far? Are we okay with acetylcholine, nicotinic receptors? Pretty good. Okay. So in question 6, so I think he usually goes over myasthenia gravis, like talks a little bit about it. Yes or no? Nod your head, shake your head if this is not related to course. Loosely related? It's in one slide. Is it green checked? He usually goes good, like check this if it's good. We don't remember. It's bolded. We'll go over it anyway. Okay. Well, I kind of gave you a little information about what it is. So a patient with myasthenia gravis is prescribed neostigmine or an acetylcholine esterase inhibitor, and we want to know what that does. So if we look back to what acetylcholine esterase does, acetylcholine esterase is what's breaking up acetylcholine. So we can kind of use that information to figure out what acetylcholine esterase is going to do. So A says it's inhibiting the enzyme that produces acetylcholine, which is not true because what produces acetylcholine is CHAT, or choline acetyltransferase. And it's not a choline acetyltransferase inhibitor, so that one's not right. B says it causes more acetylcholine to be released into the synaptic cleft. Not exactly. I think that's actually a toxin that may or may not talk about that makes the acetylcholine just be released more and more. I think that's black widow spider venom. C says it acts as an agonist for acetylcholine, which is not really true. Nicotine would act as an agonist, or if we're looking at muscarinic receptors, muscarine would do that. D says it blocks cholinergic receptors, so that would be acting as an antagonist. So that's not really true either. Our antagonist was curare. And then E was blocks the enzyme that degrades acetylcholine, and that kind of makes sense by its name. Acetylcholine esterase inhibitor inhibits the enzyme that's going to break up acetylcholine. So even if you don't know too much about myasthenia gravis, knowing kind of the term inhibitor and what acetylcholine esterase does will kind of give you what that is. So the answer for this is E. Then we can look at muscarinic receptors. So these are G protein coupled receptors. G protein coupled receptors are known as metabotropic receptors. So these are slower acting than ionotropic, and they have longer lasting effects. So it's slower to start and lasts longer. So you're usually going to find this in the parasympathetic nervous system, between the postganglion and the effector cell. This is where that's happening. So these receptors are going to be on the effector cell in parasympathetic. Specifically, the example usually given is the heart. There's also like the sweat glands and some other things for parasympathetic. Maybe I'm thinking of norepinephrine. I don't know. Anyway, so the agonist would be muscarine, which is the name, what's it named after. So this mushroom is muscarine. This is the little formula. And then atropine is its antagonist, which is shown here, atropine. So this is for the muscarinic receptors of acetylcholine. They specifically have muscarine as an agonist, atropine as an antagonist. For nicotinic receptors, we had nicotine as an agonist and curare as an antagonist. Okay, so this is what occurs at a muscarinic receptor. Specifically the one in the heart. So the action potential occurs on the neuron, voltage gated channels open, calcium flows in, neurotransmitters are then released from the vesicles. Then the ACH is going to bind to the G protein coupled receptor. So it's binding to a receptor that's next to a G protein. GDP is dissociating, and GDP binds to the G alpha subunit of that G protein. And then the G gamma and beta subunits are going to be the ones that are activating a potassium channel. When they activate the potassium channel, it opens. Potassium is going to efflux or flow out of the cell and cause that hyperpolarization, because positive is leaving and it's more negative inside, so it's making it more negative, or hyperpolarizing in this case. So here's like a picture representation of what's going on. So acetylcholine binds to the muscarinic receptor, or the G protein coupled receptor. These are the G proteins. We have the different subunits. It doesn't really show DDP and GTP that much because it's not, I guess, as important as the other ones we're going to see. But the beta and gamma subunits are the ones that are going to be activating this ion channel. The alpha subunit can go activate something else probably, but we really focus on the potassium channel because this is causing the hyperpolarization. This is usually in parasympathetic, so this is going to probably slow down the heart rate as compared to sympathetic, which will speed that up. Okay. So we have a sense. So it's kind of so we have a patient undergoing muscarine poisoning and is given atropine, and we want to know what effect will this drug have. So we need to know kind of what muscarine is, what muscarinic receptors are, and what atropine is. So A says it acts as an agonist on muscarinic receptors. So muscarine is the agonist for that cholinergic muscarinic receptor, so that's not true. B says that it enzymatically breaks down muscarine, which also not true. Not sure what enzyme does that, but we didn't really go over that at all. C says it inhibits the enzyme that produces muscarine, which is not true either. D says it acts as an antagonist on muscarinic receptors, and that is true. So atropine is an antagonist for muscarinic receptors. Muscarine is the agonist. Acetylcholine is usually the transmitter. So the answer for this one is just D. It's acting as an antagonist. Okay. Do we have questions on norepinephrine or muscarinic receptors, inotropic receptors? Are you good? Are you good? Okay. How much time? We're good. Cool. There you go. Okay, so now we're talking about norepinephrine and epinephrine. They're really similar in structure, so they both kind of activate these adrenergic receptors. It's also called noradrenergic receptors, but we usually just call them adrenergic receptors. So norepinephrine and epinephrine are also amines, just like acetylcholine, so they're being made in the axon terminal versus the soma. And then norepinephrine, epinephrine, these adrenergic receptors are G-protein coupled receptors or metabotropic receptors. So there's no inotropic receptors here. So we just have 4 different G-protein coupled receptors, alpha-1, alpha-2, beta-1, beta-2. And we see these norepinephrine or adrenergic receptors in the sympathetic nervous system between the postganglion and the effector cell. Yeah. Yes? Answer the last question. We want to know what effect atropine has. So atropine is an antagonist on the muscarinic receptors. So the answer is D. D. Because atropine is the antagonist, muscarine is the agonist, acetylcholine is the neurotransmitter. We don't really know what produces it or breaks it down. We didn't really go over that. But that's okay because we know the right answer and we don't have one with multiple answers. But yeah. Other questions on acetylcholine? Are we okay? We good? Okay. So back to adrenergic receptors. But we usually find this in sympathetic. So we'll go over the different types of receptors, a little more detail. But first, we go a little bit on how to make epinephrine and norepinephrine. So it comes from tyrosine, which is an amino acid that's then converted to L-dopa, which is then produces dopamine. And then dopamine is then converted to norepinephrine, which can then be converted to epinephrine. So the only difference between norepinephrine and epinephrine is this H versus a methyl group. So both not very similar in structure. Just like the only difference between dopamine and norepinephrine is this OH versus an H. Okay. So the first norepinephrine, receptor or adrenergic receptor is alpha-1 that we'll talk about. So the norepinephrine is going to bind to the G-protein coupled receptor, specifically the alpha-1 receptor. So alpha-1 is referring to the receptor, not the G-protein. It's referring to the receptor that's coupled to the G-protein. Because then GDP will dissociate and GTP will bind to the G-alpha subunit. So this alpha is not the same as this alpha. They're not talking about the same thing. This alpha is referring to the receptor, this one's referring to the G-protein subunit. It looks a little bit better with the picture, but we'll go through the words first. Then the G-alpha subunit is going to activate phospholipase C, also known as PLC. Phospholipase C is going to then catalyze the reaction of PIP2. I think it's called phosphoenolpyruvate, possibly. I'm not sure exactly what the names are. And it's going to produce IP3 and DHG. I think that's diaminoglutamic acid. But you might have to remember or notice what the long-handed version of the words are and identify them as the abbreviations, possibly. I don't know if we'll go that in depth. Who knows? But we get IP3 and DAG, and then IP3 is going to open calcium channels in that endoplasmic reticulum or sarcoplasmic reticulum, depending on if we're talking about smooth muscle or a different cell, because smooth muscles usually has the SR. But anyway, calcium's housed in that, so we get calcium from there. We're also kind of getting calcium from external from the cell. And then calcium and DHG are going to activate protein kinase C. So PLC catalyzes PIP2 to IP3 and DAG. IP3 is opening calcium channels. Calcium and DHG are activating protein kinase C, or PKC. And then we're going to get smooth muscle contraction from this. So let's look at a picture form of this. So we have norepinephrine binding to the alpha-1 receptor. Then the alpha subunit of the G protein is what's going to activate phospholipase C, which is an enzyme or a protein kind of in this cell membrane. This protein, phospholipase C, is going to catalyze the reaction of PIP2 to IP3 and DAG. IP3 is allowing calcium to come out of the endoplasmic reticulum, or ER, and DAG is going to help activate protein kinase C. And kinase is just phosphorylate proteins, so that's why they call it phosphorylate proteins. So protein kinase A or protein kinase C are both going to phosphorylate proteins, but protein kinase C is activated with DAG. Also, I think calcium helps too. So this one's like one of the more involved ones that we'll see. I think alpha-1 and the beta receptors are a little simpler. So if you got this down, the other ones will be fine. Okay, so we have another question. So question 8 says, which of the following is true concerning alpha-1 adrenergic receptors? So A says IP3 catalyzes the reaction of PIP2 to DAG, which is not true because PIP2 is the one that's catalyzing the reaction, and we're creating IP3 and DAG. So A is not good. B says the G-alpha subunit activates adenylate cyclase, and we actually haven't talked about adenylate cyclase yet, so this is not true either. We said nothing about adenylate cyclase in alpha-1. C says PIP2 activates protein kinase C, which is not true. PIP2 is going to go interact with the endoplasmic reticulum and allow calcium to go into the cytoplasm, which is a little bit different. So DAG is usually the one that's kind of credited for activating protein kinase C, although calcium helps a little bit. So C isn't good either. D says IP3 and DAG are second messengers, and that one actually is true. So we have, yeah, we have a true, yay! Because E says all of the above, and pretty much if we can say 3 of these 4 are false, then we can kind of say, okay, well, this one's true at least. So IP3 and DAG are the second messengers in this cascade. So if we look at it, we have neurotransmitter, receptor, we have G-alpha subunits interacting with the enzyme, and then we have these second messengers that are then going to activate things and cause an actual response in this neuron or cell. Okay, so question number 8. The answer is D. Okay, then we can talk about alpha-2 receptors. So this one's a little shorter. So norepinephrine is going to bind to the G protein coupled receptor, also known as alpha-2. So a different receptor. GDP is going to dissociate. DDP will bind to the G-alpha subunit. So same thing happening here too. And then the G-alpha subunit is going to inhibit adenylate cyclase, or adenylyl cyclase. Sometimes they'll put YL and not put this part. Same thing. Also known as AC. And then this is going to decrease intracellular camp, or cyclic AMP. So this one's a little bit shorter. Let me look at the I don't have a picture of the mechanism. Oh wait, I do. It's just kind of put together with beta. So we'll just go over beta first, or maybe we'll actually look at alpha too. So alpha 2, we have norepinephrine binding to alpha 2, and the G-alpha subunit is going to inhibit, so that's why it has a negative sign, adenylate cyclase. So it's not going to make camp when it has the negative sign here. This one stops it from working. But if we look at the beta receptors, so beta 1 and 2 are really similar. The difference is really where they're located. I think beta 1 is usually for the heart, but that's a little more information than you really need to know. So one says norepinephrine is going to bind to the G protein coupled receptor, which is still true. GDP dissociates, GTP binds. The G-alpha subunit is now going to activate adenylate cyclase. Adenylate cyclase is then going to catalyze the reaction of ATP to cyclic AMP, which cyclic AMP is now going to be that second messenger. That increase in cyclic AMP is then going to help activate protein kinase A, which is another kinase, so it's phosphorylating things. And we can kind of see that in this picture over here. So norepinephrine binds to the beta receptor. the alpha subunit is going to have a positive effect, or it's going to activate adenylate cyclase. Adenylate cyclase is going to take ATP and convert it to cyclic AMP, and then CAMP is going to activate the protein kinase. So it's a little bit shorter than the other ones, although alpha 2 is the shortest. Okay. So we have another question. Question 9 says, which of the following is true concerning CAMP? So A says, adenylate cyclase catalyzes the reaction of CAMP to ATP. That one isn't true because adenylate cyclase or adenylyl cyclase is actually catalyzing the reaction of ATP to CAMP. So we're producing CAMP, we're not using CAMP to produce other things. B says intracellular CAMP increases the activation of alpha 2 receptors. And that is not true because alpha 2 receptors are what is inhibiting adenylate cyclase from creating CAMP. So we're not going to increase CAMP with alpha 2 receptor activation. So B is not good either. C says CAMP stands for cylindrical ATP, which I don't know, that's not right either. C is cyclic, so like a cyclic, not cylindrical ATP. So that one's not good. D says it activates protein kinase A with the activation of beta receptors, and beta receptors are going to cause CAMP to activate protein kinase, well, specifically protein kinase A. So D is true. D is the correct one. E was just none of the above, but D was right, so we can't just pick that. Okay. Then we can look at a few other neurotransmitters that aren't, I guess, as heavily gone over usually. So glutamate is excitatory, so it's going to produce EPSPs. It's going to depolarize when it binds to its receptor, usually, and it's going to be the primary excitatory neurotransmitter in the brain and the spinal cord. Gamma aminobutyric acid, or GABA, is going to produce iPSPs, and that's usually most abundant in the brain. And then glycine is going to be producing iPSPs primarily in the spinal cord. So GABA and glycine. Glycine's for spinal cord, GABA's for the brain, so B for brain. Then we can look at some of the glutamate receptors. So glutamate has three receptors primarily, but we really just go over two of them in depth. So the first one is AMPA receptors, and the second one is the MDMA receptors. So the AMPA receptors are a little more straightforward. So glutamate's binding, and then sodium is allowed in, and that causes a depolarization. And NMDA receptors are a little trickier because they have that magnesium blocking it. So not only does it need glutamate to bind to the receptor, it also needs a voltage change or a depolarization for that magnesium block to kind of let loose and get out of there. So AMPA receptors are usually used to create that depolarization. So AMPA receptors bind glutamate, allow sodium in, cause a depolarization, and then the NMDA receptor can now bind with glutamate and release the magnesium and allow calcium to flow in. Calcium is then going to activate calmodulin or CAM kinases, and then this could lead to other messengers, and it could also lead to insertion of more AMPA receptors, which is going to lead to a faster response. So mainly when they talk about long-term potentiation and long-term memory type stuff, they talk about NMDA and glutamate receptors. There's also kainate receptors, but I think they don't really go over too much about them, but just know that they bind glutamate. Okay, and then we can go over stuff about the sensory system. So for the sensory systems, this is kind of where I need some information. I know they went over taste. I know they usually go over auditory and language stuff. Did they go over, or did he go over olfaction and vision, or no? Olfaction, yes. Vision, no. Okay, then we skip over vision. Because I put all of it in case, because I'm not sure exactly what you guys touched on. I know vision and auditory, not vision, sorry, taste and auditory were definitely there, but of course olfaction apparently is there as well. Also somatosensory mechanical receptors are usually on there as well. So we'll just skip over the visual part, because we don't need it. But this kind of gives you an idea of what's happening in olfaction. So this is kind of the olfactory bulb and inside your nose, kind of what's going on here. And you can see that not only does olfaction have neurons that go to the thalamus, but they go directly to the amygdala area and the hippocampus area. So amygdala area, hippocampus area, without going to the thalamus first, which is one of the, I think there's two of them. Also, like the optic nerve doesn't also go through here. Yeah, there's only like a few types of nerves that don't go through the thalamus before touching on other areas of the brain, and the olfactory bulb or nerve is one of those special ones that can go synapse with other parts before going through the thalamus, which is usually where everything is kind of sent off to different parts of the brain. So that's why sense of smell can tap into memory or tap into fear or other kind of emotions and be quicker. So then we can look at a little more closely. So odorant molecules going in, and we have our olfactory neurons coming in this way, and then we can look at the signal transduction, so even closer than this. So this is the closer version of it and actually what's going on. Did they go in depth enough into the signal transduction of olfaction, or? The light cyclase, okay. So they kind of talked about it a little bit. So we have another G protein coupled receptor, so the odorant molecule is binding. The alpha subunit is activating adenylate cyclase. Again, adenylate cyclase is converting ATP to CAMP. And then CAMP is now, instead of activating like a protein kinase, it's going to activate this cation channel, which is going to allow calcium and sodium in. The calcium is then going to activate this chlorine channel, and chlorine is going to rush out. So we have different channels being activated. So it kind of starts out similar to beta receptors, where alpha subunit activates adenylate cyclase, converts ATP to CAMP, but instead of CAMP activating a protein kinase, CAMP is going to activate an ion channel, which is then going to activate another ion channel. So these are G protein coupled receptors that are then going to activate ion channels primarily for their effect. Okay. Yeah, so to like make sure you don't get these things kind of mixed up and confused, I would kind of think about making a chart to compare what receptors, what enzymes they're activating, and then what kind of effect they're going to have. So you can see like the things that do use adenylate cyclase, the things that inhibit it, the things that don't use it at all, and that sort of thing to kind of keep them separated and make it less confusing. Okay. Also, drawing them helps as well. So not just seeing them, but also seeing animations and also drawing it out yourself is very helpful. Then we kind of have, I thought this was a cool picture for the pathway for taste, and it's showing you your cranial nerves and how your facial nerve is for those anterior 2/3, and your glossopharyngeal is for the anterior 1/3 for the sense of taste. It also helps with, then you have your other motor neuron for your glossopharyngeal. And how it goes into your brain. So these are from cranial nerves, so that's why they're synapsing in, I think it looks like the pons medulla area, because this is the midbrain, and it's going to go up and go into the cortex through the thalamus. But we'll go to take a closer look at taste. So we have 5 tastes. So for glucose, or for sweet, we detect glucose. Salty, we're detecting sodium ions. Umami is glutamate. Bitter is quinine, I think. Quinine. And sour is those hydrogen ions, or H pluses. Okay. So then we have a little closer look at what's going on. So in salty, we have sodium going through the ion channels, active. They're probably going to depolarize and open these voltage gating calcium channels, and that's going to release neurotransmitters. So this is the taste bud type receptor, the specialized sensory receptors, and they don't look like normal neurons, but they release neurotransmitters, and that's kind of what makes them neurons. Bless you. Okay. So this is the taste receptor cell, and then we have the primary afferent neuron, which is going to receive the neurotransmitter and then produce an action potential if it's excitatory. Okay. And then for sour is over here. Sour is blocking potassium channels. So if potassium can't get out, that's causing kind of like a depolarization, or enough of a depolarization to open calcium channels. So it's a different way for it to do that. So blocking potassium from leaving. So this is allowing sodium in, this is blocking potassium from going out, but they're both activating these voltage gating calcium channels. And that's going to then, calcium helps release neurotransmitters into that synapse. So salty is allowing sodium in to depolarize, sour is blocking potassium from leaving to depolarize. Then we have sweet and bitter. So these are using metabotropic receptors instead of ionotropic receptors. So we have the glucose binding to its receptor. We're going to have the activation of adenylyl cyclase. So ATP is going to CAMP. CAMP is then going to activate the protein kinase, which is going to act or just close these potassium channels. Activates the protein kinase and closes the potassium channel, and then that's how it allows calcium in to depolarize the cell. So sweet is kind of like the sour in that it's stopping potassium from leaving and opening a channel, but it uses a G protein to do that instead of hydrogen ions to do that. So it's using a G protein to create CAMP to make this protein kinase close the potassium channel. That's how sweet and sour are kind of similar in their mechanism, though. And then neurotransmitter release, the afferent neuron, is then going to send that message to the brain. For bitter, we also have a metabotropic receptor, but now we have PLC being activated, or protein lipase C. Oh, yes. Oh, that's nice. Thank you for telling me. Okay. Well, some people keep an eye on that, maybe. Got me, cool, cool. Okay, I don't get those alerts. Maybe I do. No, I don't get those alerts. But we're not in the library, so positive note, positive note. Not funny, but okay. Okay. We're almost done with our review, so we'll just continue. We have like 20 minutes, 25 minutes, and then we have questions at the end if you like. But yeah, we just have senses left, so we'll go over that. Yeah, okay. But so bitter is kind of like the other neurotransmitter transduction thing we had, where we have phospholipase C catalyzing PIP2 to IP3, and then IP3 is opening calcium channels inside the cell, so the endoplasmic reticulum or the sacroplasmic reticulum. And then calcium is releasing neurotransmitters. So the big thing is calcium is what's releasing neurotransmitters. So if there's one thing to take away, it's calcium causes the release of neurotransmitters. Then we said vision isn't really on there, so we'll ignore it. Then we kind of have some areas to think about for auditory. So we have Broca's area, so that's understanding not understanding language, so it's it's the motor movements of language, being able to speak and talk clearly. That's why it's near the motor area, the primary motor cortex. Then we have the sensory motor cortex, and that's kind of where we're going to find, or not where we're going to find, but we have primary auditory in the area on the temporal lobe. And then in the associative areas, we have Wernicke's area. So that's understanding speech and being able to know what someone's talking about. And then it uses the associative areas as well. So Wernicke's, you need it to understand language. Broca is to speak and use language, or speak and create the language. Wernicke is to understand it. So when you have Broca's aphasia, you can't really speak very well. Things are jumbled, but you understand what you're trying to say. You know what you're trying to say, but speaking is just not very good. In Wernicke's area, or Wernicke's aphasia, the receptive aphasia, the person kind of doesn't know what they're talking about. Like, they don't realize that they're speaking in nonsense and long sentences, but they still kind of use grammar and syntax. They just kind of input words that shouldn't be there or make up words. They don't really know what they're doing. But they sound like they would be talking normally if they were using the correct words. So then we have a little bit about the ear, the auditory transduction. So sound is coming into the eardrum. It's going to go against the ossicles and move them mechanically against the oval window of the cochlea, and then that's going to produce pressure because inside the cochlea is a bunch of fluid. So we're going to have pressure on the fluid, and pressure on the fluid moves the basement membrane, which is then going to move some hairs. So this is all really mechanical in what's happening in the ear. So if we get a closer look, this is inside the cochlea. So we have our basilar membrane, and we have our hair cells. So the inner and the outer hair cells. If we go one step closer, we can look at an individual hair cell, and we can see those mechanoreceptors. So literally when hair cells are moved, they pull the receptors open, and potassium is allowed to flow in. So the difference here is that potassium is causing a depolarization effect in the hair cells. So usually when potassium channels are open, potassium flows out and we hyperpolarize. But in the hair cell, the inside of our cell, of a hair cell, is okay, so the paralymph and endolymph. So if we look back over here, so inside of our actual cochlea is going to be the endolymph inside these canals, or inside of this canal specifically. So can we see that canal specifically? Kind of. So inside this one we have endolymph, and inside the other ones, so we have endolymph here and paralymph over here. And that's kind of what's affecting our concentration. So what's usually high potassium inside the cell is now the opposite. We have low potassium inside, high sodium inside in our hair cells. So it's going to be opposite of what we would usually think for regular cells. So when we have potassium channels opening, instead of depolarizing and potassium rushing out, we have potassium rushing in and a hyperpolarization, or not a depolarization, sorry, of the cell. That depolarization opens calcium channels, and calcium is releasing neurotransmitters, which is kind of the overall thing that doesn't change. So although the concentrations of potassium and sodium change in hair cells, the effect of calcium causing the release of neurotransmitters is staying the same throughout. Okay, and then we have question 10. When asked a question, a patient gives a long answer with no meaning. The patient speaks using grammar and syntax and seems unaware of the long and incoherent speech. So which of the following is a likely diagnosis? So we have A is Broca's aphasia, B is expressive aphasia, C is receptive aphasia, D is retrograde amnesia, and then anterograde amnesia. So aphasia is not being able to find, like, no words, kind of, in a way. And then amnesia is just forgetting and, like, memory loss type thing. So retrograde would be not being able to remember anything before a certain time, and terograde would be not being able to make new memories. Receptive aphasia has to do with not understanding what's being received, or not understanding the information, or not being able to, like, form coherent meaning. And expressive is how you mechanically express your thoughts. So Broca's aphasia and expressive aphasia are actually the same thing, and receptive aphasia and Wernicke's aphasia are the same thing, or they're different names for the same thing. So if the patient can use grammar and syntax and just kind of doesn't give answers with meaning, that's not really a problem with the mechanics of language, it's a problem with the understanding. So that's a receptive aphasia. That's Wernicke's aphasia. So question 10 is C. Okay. Yeah, then we have somatosensory about the touch and the mechanoreceptors. Yeah.
If the patient not speaking something,
The patient gives a long answer with no meaning. The patient speaks using grammar and syntax and seems unaware of their incoherent speech. Oh, incoherent, maybe that wasn't a very good way of saying that. So the point was that they can answer and they can talk back, but they're just not making sense in what they're saying.
so what would be the actual function?
Yeah, Broca's is like you're tongue-tied and they can't, like, create the words. But if you, like, help them with the words, they're like, yeah, that word, and they can kind of give you meaning if you kind of help out. But with Wernicke's, you can help them with the word, but they don't realize what you're doing. Okay. Other questions? Okay. So then we have kind of the graph of, I think they call it the homunculus, which kind of shows you how much information, or how many of the neurons on the cerebral cortex are used for specific body parts. So I think the left is motor. So the left is motor, and the right is talking about the sensory map. So motor and sensory, they're kind of similar in how big, like, the face is and versus, and the hands are very sensitive, and very, like, fine motor skills are allowed with that. So they're very similar, but slightly different. So just knowing kind of that's where you would find fine motor skills and where you would find very sensitive areas. So I think talking about, like, the 2-point touch is one way of figuring out sensitivity of how many neurons there are innervating a certain area. So, like, comparing the 2-point touch on your face versus on your back, the back would have, like, a larger distance between where you can figure out 2 points are touching, versus on your face where really close points would be noticeable as 2. So that kind of has to do with the sensitivity and how much innervation is there, and how much in your brain accounts for that. I think they also did that experiment, I think it was with the monkey hands, where they were taking away fingers, and in the brain they could actually use the information that was used for that part of the hand and replace it with something else. They can kind of learn to use some of their portions of their brain for other things, and that kind of showed how plastic the brain is. Kind of like when someone's blind, they don't just not use their visual cortex, they use it for other things, so it gets used in a different way when you don't have information coming from those senses. So the brain is plastic in that sense. So although this is kind of the setup of what it would look like, but if anything happens, any, like, sort of damage where you lose a limb or a sense, that can kind of be picked up by other areas. It's usually by, like, surrounding areas, but it probably depends on how old the person or animal is, and then other factors as well. Do we have questions on the homunculus? Are we okay? Okay. And this is kind of the tract of what's happening. So we'll have sensor information from the free nerve endings, talking about pain, and then temperature. We'll also have information from the muscle fibers themselves for proprioception to figure out like kind of where your body is in space. And we're going to also have kinesthetic receptors in the joints. It also helps with the proprioception as well. So these are kind of for that, to figure out how your body is moving, and this is more where pain is, and also those mechanical receptors to figure out pressure and temperature as well. So this is all going up through the spinal cord and going through the thalamus. So this proprioception type information needs to go to our cerebellum for balance and coordination type things, and we also go to the motor cortex for trying to actually do different movements. We have different somatosensory nerve fibers for whatever we're working with. So for alpha or A alpha, so we have A and C. We usually have C fibers are usually pain. They do temperature, pain, and itch, and they're unmyelinated, so they're not as quick for pain sensation. So these are more the dull pains that you're going to feel. Compared to the myelinated pain receptors, which is the alpha or the A delta. So A delta pain receptors are going to do pain and temperature, but this is the quick one. So when you get burned at really high temperatures, or you have like a really like a cut, something that's really painful and very serious and needs your attention right away, those are going to be the myelinated ones, versus the duller pains that don't get your attention as quick. Those are unmyelinated. Then we have mechanoreceptors of the skin, so those are the beta receptors, and then we have proprioception of skeletal muscles. So in our joints and also in those muscle spindles, that's where we'll have the A alpha muscle fibers. These are the thicker ones and heavily myelinated, so they have very fast transduction speeds. I think these are the speeds down here. I don't know that the graph kind of cut them off, or the table cut it off, but these are the thickest and the quickest, and then they get thinner and thinner until they're unmyelinated and very small diameter. And we know from way back in the beginning, the larger the diameter, the easier it is to flow along the axon, so the decreased resistance along the axon, and then the more it's myelinated, the increased resistance across the membrane, the less likely things are going to leak out. So it's kind of like tape around a leaky hose, or having a very large diameter hose versus a very small one. So like the difference between a firefighter hose and like a home garden hose. One is going to be more efficient at water traveling through, or faster, and the other one's going to be a little more slower, or a lot slower. So those are fibers, where you find them, and then we can look at our mechanoreceptors in our skin. So we have Pacinian corpuscles, so in the deeper skin, versus Merkel discs, which are higher up, and Meissner corpuscles, which are also higher up. So the ones that are more disc-like are usually better at detecting pressure for long periods of time. Do I have that graph? I don't have that graph with me, dang it. And then we also have, where are they? Ruffini endings in the deep as well. So Ruffini endings and Pacinian corpuscles are deeper in the skin, versus these Merkel disc and Meissner corpuscles. So kind of knowing how deep they are in the skin and how they affect what kind of pressures are good for them will be helpful. So those mechanical receptors are the beta receptors, or the A beta type fibers. So A beta for that. Then the temperature and pain, so these free nerve endings are going to be either delta or C fibers, depending on if they're quick pain or non-quick pain. So this looks like unmyelinated because it doesn't have any myelin on it. Possibly. So we have all of these in the skin. This is mainly in the muscle, or in the muscle or the joints for proprioception. This is more in the skin for pain, temperature, and pressure sensing. And that's, I think, all I have for you guys. So we ended a little bit early. But if you have questions or want to go over other stuff, we can do that too. Because I'll be here till 5:30, technically. Wish I had more questions prepared. We can go through the other questions if you missed them or missed the beginning.
I was sorry for hanging out. Questions on 6, 7.
6 and 7?
Yeah.
Cool. Okay. Okay, so 6 was about myasthenia gravis. And we said a patient with myasthenia gravis is prescribed neostigmine, and we said that that's an acetylcholine esterase inhibitor. So that was like our bigger hint of what this is going to do. We want to know what the effect is going to be. So an acetylcholine esterase inhibitor, we think is probably going to inhibit acetylcholine esterase. And then if we back up, we can see that acetylcholine esterase is the enzyme that helps break up acetylcholine into acetic acid and choline. So if we look at our answer choices, A says it inhibits the enzyme that produces acetylcholine, which is not true because chat is what produces acetylcholine, or choline acetyltransferase, and it doesn't inhibit that, or else it'd be acetylcholine transferase inhibitor, not acetylcholine esterase inhibitor. So A is not good. B says it causes more acetylcholine to be released into the synaptic cleft, which is not true either, so it's not like making more calcium happen and more vesicles being releasing neurotransmitters, so that doesn't make sense. I think one of the toxins does that actually. So if you look at the toxins for acetylcholine, that might be one of the things. C says it acts as an agonist for acetylcholine, which is not true either, because the agonist for acetylcholine for nicotinic receptors is nicotine, the antagonist is curare. D says it blocks cholinergic receptors, so blocking cholinergic receptors would be an antagonist type activity, so that would be like curare. And then E says it blocks the enzyme that degrades acetylcholine, and that one is true. So acetylcholine is degraded by acetylcholine esterase, and it blocks or inhibits the acetylcholine esterase, since it's an acetylcholine esterase inhibitor in its name. So with that one, you don't really need to know that much about myasthenia gravis or neostigmine, as long as you know kind of what an inhibitor of acetylcholine esterase is. Okay. And then 7, so 7 about muscarinic receptors. So this is, again, still an acetylcholine, but now the metabotropic receptor for acetylcholine. So 7 says a patient undergoing muscarine poisoning is given atropine, and we want to know what the effect will be for atropine. A says it acts as an agonist on muscarinic receptors. So for muscarinic receptors, muscarine is the agonist, atropine is the antagonist. So it's not going to act as an agonist. That's what muscarine is. And plus, that would make the problem worse if it's acting as an agonist for something that's causing problems. B says it enzymatically breaks down muscarine, which we don't really know if that's true or not. We don't really know what the enzyme that breaks that down is. C says it inhibits the enzyme that produces muscarine, which we also wouldn't really know from our notes because we don't know what produces muscarine either. We just know that we have a problem. We're using atropine to solve it. And then D says it acts as an antagonist on muscarinic receptors, which is something we do know. Muscarine is the agonist, atropine is the antagonist, so it's going to block those receptors so muscarine can't bind. And acetylcholine is the neurotransmitter. So the answer for this one was D. It's acting as an antagonist. You're welcome. Do we have other questions? Are we okay? Or questions on what I didn't go over? I know I'm going to post this on the Google Sites, and then Sam posted her review, which is a little bit, includes a little bit more of other things on it. So she has memory and some neuroscientists. I don't know, actually chapter 1 wasn't really covered heavily, actually we're tested on. So I don't know how much information he has on that, probably not much. But memory stuff and those experiments with those neuroscientists are on there as well. If you want more experience, yeah. Okay, then yeah. So on the other slideshow, which is on the Google site already, so if you want to look at what the Google site website is, because if you try to search it on Google, you're not going to find the UCF Study Union one. You're going to find the SI one, and it's not on the SI school site. So you can either go to this link, or you can go to the SARC Facebook page, and that will have the link. So SARC Facebook page, or this one right here. If you want to see this review again, it's going to be recorded and posted on the YouTube page for SARC. Also, we have ones from previous semesters, previous study unions that are also posted on the YouTube page for SARC. So you can either watch the one that Sam and I gave last semester, or you can even watch the one from the previous SARC tutor for Neurobio. Joan is on there as well, and he has a really good review as well. But his focus more on Fernandez teaching versus Chang and Bossy teaching. So maybe there's some stuff that's missing versus some stuff that's added on. But it's a really good review overall. So I would recommend that one. So that one's on the YouTube page for SARC. So if you go on youtube.com and look up UCF SARC, youtube.com, and look at UCF SARC, you will find the other reviews. Okay. And then I'll post this one on the Google site so you can look through the questions again if you want, and I'll actually bold the answers so you can see them maybe, or I guess I'll put the answers at the end. I'll put it on the slide at the end so you can review if you would like them. But other than that, other last minute questions that we have, either on this material or stuff I didn't cover, or if not, then good luck on your exam. Hope you do well. Be safe, whatever's happening. I don't know what's happening. Something happened. There was a threat, or there wasn't a threat? Okay.